Welcome to DNASTAR LabVIews. My name is Katie Maxfield, and I'm joined today by Dr. Richard Buick, the Chief Technical Officer at Fusion Antibodies in Belfast. Thanks for joining us today, Richard.
Thanks for inviting me, Katie.
So to start off, can you tell us a little bit about your work at Fusion Antibodies?
Yep, certainly. Fusion Antibodies is a CRO that specializes in antibody development, antibody engineering, and antibody humanization, and developing recombinant antibodies going right through to cell line development for mostly therapeutic applications.
And can you talk a little bit about the antibody humanization service that you work on?
Yeah, humanization is the process of taking a mouse antibody sequence and making that mouse sequence, amino acid sequence, more human so that it's less immunogenic in patients. We would start with the mouse sequence. We can actually sequence antibodies as well. But once we have the sequence, we'd compare that sequence to a database of human antibodies, usually millions of human antibody sequences, and narrow that down to about 200 mature human sequences. These are human antibodies that have been developed within a human being, so not from phage display or recombinant techniques are designed. They're actually naturally somatic mutated human antibodies. And from those 200 sequences, we would then search for particular human frameworks that have key residues in common with the murine antibody, and then perform traditional CDR grafting from the murine sequence into the human frameworks. And then we end up with a panel of about 10 to 25 humanized antibody variants based upon the original mouse sequence.
And why is it so important to have humanized antibodies?
Well, it's because of immunogenicity, really. If you were to inject a mouse or any other species antibody into a human patient, the patient's own immune system would recognize that as being foreign and would then destroy it. Whereas if you humanize the antibody, actually to make the mouse antibody or other species antibody more human, then it fools the human immune system into accepting it as being human, and then you can give repeated doses without the patient immune system getting rid of it.
And so what would you say are the biggest challenges in antibody humanization?
It's getting that balance between as being as human as possible, but also retaining the function of the original mouse antibody sequence. I'd say the main challenge is being able to have a humanized antibody which has exactly the same characteristics as the mouse antibody, but of course is non-immunogenic. And that's the whole goal of the humanization.
Right. So switching gears a little bit to how DNASTAR fits in, can you talk a little bit about how you've used NovaFold for your service?
Yes, whenever we perform the humanization, we would end up with, say, on average, 16 humanized variants. And we would use the NovaFold software to model each of those variants and then compare that to the original mouse. And because we can overlay the sequences, overlay the actual 3D models, we can see exactly how the humanized variant has changed in comparison to the mouse. And we can see if there's key structural parts of the antibody are exactly so overlaid on top of each other, or if there's been some variation between them. And of course then that would translate into a change in the function of the humanized variant.
And so what feedback have you gotten from your customers about the NovaFold results that you provide?
It's been very positive, really. A lot of the customers have said, rather than just looking at the images, they can actually almost predict which ones are going to perform the best. And we can see this particular variant definitely looks as if it's got the closest structure to the original murine antibody. In fact, there was one client actually had crystallography structural data for the murine antibody previously, and they were able to tell us that the NovaFold structures were almost identical to the crystallography structures that they already had. And that was obviously just NovaFold's entirely just in silico predictions.
Wow, that's very cool.
It is, yeah, so it was a good good confirmation of the structural predictions.
Definitely. Well, thank you so much.
No problem, thank you.
To see more interviews with DNASTAR users, please visit our website, DNASTAR.com. And to have your work featured on DNASTAR LabVIews, contact us at DNASTAR-info@DNASTAR.com.